Well, good afternoon, everyone, or good morning for some of you, maybe. I'm pleased to talk to you today about cell wall polysaccharides, impact on textural properties, and how to study them, or rather, how we try to study them in INRAe. So, why did we choose to focus on cell walls? Well, Firstly, some of you are already studying other aspects of biochemical determinants of textural properties, such as starch content, dry matter, or amylase. And what is interesting in your findings is that it explains some of the textural properties, like firmness, but it cannot fully explain everything. For example, mealiness is kind of hard to explain. In INRAe, we did research in the past on non-starchy products like apples, and this research showed an impact of pectin on textural properties in both raw and cooked products. And because of that previous experience, we thought it would be a relevant area of research for RTB foods. Moreover, 2 weeks ago, the GHI showed us the work on cooked potatoes and the relation between pectin methylesterases and texture of cooked potatoes, which also indicates an influence of pectin. So cell walls in vegetables, they constitute the backbone of the plant structure and are composed of 2 main types of polymers. There are the polysaccharides, cellulose, hemicellulose, and pectin, with pectin being more in the middle lamella and in the primary acidic plant cell wall. With different areas, you see Hg for homogalacturonan, which is linear pectin, and Rg for raminogalacturonan, which is more ramified pectin. There can also be polyphenolic polymers like lignin. In yam and in plantain, there's not that much lignin, but we can find it in some RTB foods like sweet potatoes. So pectin is a polymer characterized by its polygalacturonic acid backbone. Like I showed you before, you can have linear area, which are called homogalacturonan, and ramified area with lateral chains composed of neutral sugar. Pectin play a major role in textural evolution during cooking processes. So, as I said before, you will find 2 main types of pectin in cell walls in 2 different areas. So, in the middle lamella, you will have linear pectin, which are also mostly methylated pectins. They are more soluble than the pectin you will find in the primary cell wall, which are ramified, normally you can see this, which are ramified pectins with a lower methylation degree. So it's one polymer, but you can have more ramified pectin in the primary cell wall and more linear ones and more methylated ones in the middle lamella. This will impact the texture and mechanical properties, both in raw and in cooked products. So in raw products, cell wall have mechanical properties, especially the primary cell wall, which impact texture, and soluble pectin, which is pectin of the middle lamella, ensures the intercellular cohesion. So it's partly responsible for the viscoelastic properties of cells. And what is interesting is that when you cook the products, some polymers like cellulose and hemicellulose don't change that much, whereas pectin do. These are the results of the research led by Catherine Renard. So we know that pectin is more prone to changes during cooking processes. what can be these changes? So here you have a picture of raw and cooked onions. So what does happen is that during cooking processes, especially during boiling processes, you will have a solubilization of the soluble pectins from the middle lamella, and they form viscous solution. Because they are leaving the middle lamella, there is an increase of the cell wall porosity, which leads to more exchanges with the surrounding water and a loss of turbidity. You also have an increase of the cell wall thickness, so you can see it with the onions here, but Noel Akizoe also showed it in his own research work with yam, that you can also see the same process. The low methoxypectin, which is a pectin of the primary cell wall, will go under gelation processes. It's a process that will take a longer time because it's hard to extract pectins, which are non-soluble pectins trapped in the primary cell wall. But this can happen when cooking for a long time. And there's also enzymatic activity. So pectin metylesterase can act and react with methylated pectins to demethylate them. And so what we see when we look at the global picture when cooking food is that we have an increase of viscosity linked to the solubilization of pectin and which form viscous solutions, followed by a loss of the viscosity when we cook too long, because the pectins, the soluble pectins, just go, the soluble pectin just goes into the liquid, the surrounding liquid. And we have generally a decrease of pectin molar mass. So for yam and plantain, we can mainly have two types of reaction, we think. So we can have a demethylation of all methylated pectin by pectin metylesterase. So it is active between 50 and 82 degrees. So it gets deactivated at some point, but not at the beginning. And demethylated pectin will lead to gel formation and an increase of viscosity. This can happen, but what can also happen with methylated pectin is we have beta elimination chemical reactions. This can happen because of the pH of these foods. Not all foods have such a high pH, and plantain and yam do have a pH higher than 3.5, which can lead to beta elimination when the temperature is over 80 degrees. So once we studied the literature, and we could draw some objectives. So what are our objectives in the project? For us, it is to research the impact of the cell wall and the pectin structure and the composition of the pectin on the texture of boiled yam and boiled plantain. We would like to investigate more specifically amount and structure of both soluble and non-soluble pectin, because as I told you, soluble and non-soluble pectin, which are also methylated and non-methylated pectin, do not undergo the same processes when cooking. And finally, we would like to understand the mechanism which takes place during the boiling process. To do so, we have chosen to work with well-characterized, controlled sources of yam and plantains with contrasted properties as much as possible. We do not have an analysis method which would allow us to do an analysis on raw food without any preparation, so we need an extraction step of cell walls from all products. And finally, we would like to analyze the cell walls and the pectin structural characteristics. As said before, pectin is a polysaccharide. The issue with RTB food is we have another polysaccharide which is much more present, and it is starch. So what we are so in ripe plantain or in yam, we have between 60 and 90% of starch in the dry content. So it's really too much for our analysis methods. That's why we need to distarch it. We tried using physical separation, like sieves, but it didn't work. We tried chemical extraction with dimethyl sulfoxide, and it did not work. And we finally chose an enzymatic approach using amylase to distarch our product with a two-step heating procedure, first 6 minutes at 85 degrees and then 3 hours at 50 degrees. Because we chose the enzymatic distarching, we need to treat our dry content to remove the polyphenols. Polyphenols inhibit amylase, so we cannot keep it. And then we have this preparation step, which leads to AIS, alcohol insoluble solids. And on this AIS, we apply the distarching procedure, and then we do another extraction step to remove the soluble sugars produced by the distarching. So we started to characterize some yam cultivars from Benin and Ivory Coast, and we did not see that much of a difference between the varieties in galactose nor in galacturonic acid. What we saw and what was really interesting was that we had a low to medium methylation degree. So if you remember, I told you that methylated pectins and unmethylated low methoxypectin don't really undergo the same processes. So if we do not have that much methylated pectin, we may not have that much beta elimination, leading to a textural loss. However, there was an issue with our first distarching procedure, is that it worked very well on some varieties, and on others, like Pectin, yam, we still had 50% of starch in the distarched powder. So it was not possible for us to study all varieties with our first protocol. So we tried to find another one. We chose to use 2 enzymes rather than 1, and to keep the 2-step temperature process, but with temperature of 100 degrees for the first step. This was inspired by the literature and work on cassava. So that was our new protocol with thermostable alpha amylase and amyloglucosidase. It gave quite a good result for the distarching part, because we had nearly no remaining starch. Even in Pectin yam, we had like only 4% of starch remaining. But the issue was that on our distarched powder, we only identified 30 to 40% for the Ghana yam and unripe plantain, and 60% for the Pectin yam. So we are missing something here. We thought it could be the proteins. We had it because enzymes are proteins and they could stay in our powder. So we tried to analyze how much proteins we had left, and we only found 3%. So it may be that we are eating so much that it's damaging the pectin and the soluble polysaccharides. That's why we try to lower the temperature. We got inspired by the work of the GHI on sweet potato. They have a protocol with 20 minutes, I think, at 70 degrees, and then 4 hours at 50 degrees. So we did the same, but as you can see in the graph, we still had 60% starch left, so it was not efficient enough for yam. So we recently tried flash eating, so just a few minutes at 95 degrees, and it changed the whole thing. We still need, however, to characterize the cell wall polysaccharides of these recently obtained samples to see if there is less damage done to the cell wall polysaccharides and if we identify more of its content, of the final powder content. Therefore, our perspectives for next month and next year is the finalization of a discharging procedure, which needs to work for all varieties of yams, and that's why we are working both on Ghana, which is a commercial yam that we have widely available, but also on Pectin, because we know that we had some difficulties with this variety in the past. And then we will move on to producing knowledge about the composition of these foods. And especially, we will start by characterizing pectin composition in contrasting yam varieties that Dominique Rinaldo from INRAe Guadeloupe sent to us. So we received both boiled and raw samples, which is really interesting. But these are only Dioscorea alata, so we will also be working with Benin to study some Dioscorea cayennensis rotundata, which are quite different for their cooking properties. And finally, we will characterize different plantain varieties from Ivory Coast. All of this, of course, is done in collaboration with RTB partners. Thanks for your attention.
Many thanks, Aliénor, for this very pedagogic presentation. It's a good summary of part of the activities performed at INRAe France within RTB Foods Work Package 2 on cell wall and pectin structure. And many thanks as well for choosing to put the emphasis on the different challenges faced during the protocol development for destarching and describing the different steps to overcome these challenges. This is particularly important because, as you mentioned, other labs have been carrying out similar, or let's say complementary activities within RTB Foods framework. So it's now time for questions. So please raise your hand or write your question in the chat box if you do not feel like asking the question directly to Aliénor. Any precision, any more details that you would like to have regarding a specific point addressed during this presentation, or a possible application of the protocol, of the approach on your product in your lab? Thierry, you have a question?
Yes. Hello. Thank you for a very nice presentation.
We can hear you very slow. It's very
okay.
low, sorry.
Yeah. Hello, Aliénor, and thank you for a very nice presentation. I wanted to ask if in the end you have an estimation of the percentage of pectins that you recover compared to the total pectins.
Okay, what I recover is around I recover around 30 to 40% of my discharge powder, which is like only 8% of the dry content. So 8% of the dry content for cell wall polysaccharides is quite normal. we are expecting something in this range, but the issue is that I don't identify that much. So I only identify like 3% of the dry content as pectin and cellulose, whereas I'm expecting more like 8%. So it's really, if I just look at the mass of my final product powder, I could think, okay, it's great because I have something that I expected, but when I analyze it, I don't know what are like the 60 to 70%, which are not pectin, not cellulose, no proteins.
No protein either.
Yeah, no. Well, the thing is, we analyze the protein content by two methods. We tried gel separation to see what protein we had. So we saw that we still had a little bit of the proteins we added and also native ones. And when we try to assess their concentration, well, it's soluble proteins that we can only assess, and we found about 3%. So there's two possibilities. Or the first one is it's not proteins, and the other one is that we don't fully identify all protein either, because some of them may not be that soluble and sink back and stay with the pectin.
Okay. So can it be that I don't know, the pectins, part of the pectins are depolymerized and we lose them during the washing?
Yeah, also that can be an issue. That's why we quench it with 96% ethanol to go back to 70% ethanol to try not to lose too much. We tried with 70% ethanol, and we saw that we lost more pectin.
Okay. Yeah, we're working on a similar approach on cassava, and we have very similar issues, let's say.
Oh.
Thank you.
Great, it's nice to know we are not the only ones.
Yeah.
Thank you, Thierry. There's a question from Mark.
There's also a question from Suzanne in the chat.
Yes. If you want to address Ms. Suzanne's one before.
Yeah, because I know the answer. The typical protocol for making AIS is we rinse it with 70% ethanol if it's a dry powder, with 96% if it's a cold powder. And we know it removes effectively the polyphenols, and then we rinse it with acetone. And polyphenols are really soluble in acetone. And it was not tested for yam and plantain specifically, but we have a unit for polyphenol testing in INRAe Avignon. And they are really well aware of the issue of polyphenols. What we tried also is to see if we did a pre-treatment with acetone for the removal of polyphenols, it would enhance our discharging process, which it did not. So we think that our current procedure with ethanol and then acetone is enough to remove it. And also we had quite the same result for discharging between plantain and yam. And with our actual protocol, we do not see that much difference between varieties, which is quite great, because we are expecting more polyphenols in yam than in plantain. That's what the literature told us. So we think that we remove it, but we did not test right now the amount of polyphenols in our AIS.
Thank you, Aliénor. So Mark, please, you can ask your question to Aliénor.
Thank you very much for that presentation. Great work. It's not easy at all. But my question is, and I might have missed it, but how much variation in cooking time is there between the different yam varieties that you're looking at?
We are for the varieties that have always been characterized. I'm not sure for the varieties we just received. It's not that much because in just Korea lactase there's not that much. So it's like from 15 minutes to 25 minutes, and that's why we are really looking forward to working with cayennensis for data. That's an issue. We have contrasted varieties we try to have, but there's not that much contrasted. Pectin is different, but I think it's harder to cook, but we could not fully characterize it in the first row because there was still too much starch left.
It's always easy when you've got some very large contrast to look at. Thank you.
You're welcome.
Dominique, you have a question for Aliénor, if you want to ask it directly.
Yes. Aliénor, thank you for the presentation. Very nice to see your work. And it seems that the galacturonic acid level, the content is very similar in all your samples. And the methylation is the only thing who is changing between samples.
Yeah, it is.
The question is about experience or literature review about this methylation and overcooking type in fruit and tubers. Sweet potato, potato, banana.
The methylation degree we found were in the range we expected. However, you have to be really careful with methylation degree because it's an analysis which adds on errors. I explain. If you still have starch left in your sample, it will impact the galacturonic acid Oops. Sorry. So it will impact the galacturonic acid analysis. Sometimes it gets higher, sometimes it gets lower. So it's not. So you have an error on this, and then you have the methanol analysis, which can also be impacted. So when you do the methylation degree, which is assessed by using the quantity of galacturonic acid measured and the quantity of methanol measured, you have a lot of error. That's why I stick to say no to medium methylation degree, and I did not push my luck further because we know that this number we found, they probably have a large error, a large deviation.
Okay, thank you. And the question is now, how did you characterize the cooking time? I know that Dominique Rinaldo is working on in Guadeloupe.
Yeah, we did not. So it's all Dominique Rinaldo's work.
Yeah. Dominique, you are there? Maybe you can tell us how you will evaluate.
I don't know if she's here. We talked with her and she told us that she was
She's there.
Okay.
Dominique Rinaldo?
If she...
Can you hear me?
Yes.
Yeah.
Yes. Good morning, everybody. And the cooking time was evaluated for boiled yam with using a fork, but it is always the same person who evaluates the cooking time. And I also use the variety Cabuza as a control each year, so we can evaluate if our response is good. And Cabuza from year to year the cooking time from Kabusa is about 20 minutes.
Okay, it's calibrated. And
If Agnes, if I can add a precision,
yes,
it's Dominique Rinaldo also did all textural measurements according to the SOPs determined for texture measurements.
okay, by penetrometry.
Yes.
Yes, it is penetrometry. We have used also TPA, but it doesn't work well.
Okay.
Okay, please, I have a question. This is Bolale from Nigeria. Please, I have a question.
Yeah.
Yes,
During the cooking time estimation, did you use the same volume of water? How did you standardize the cooking time measurement, the procedure? Hello?
Yeah, this is a question for Dominique Rinaldo, I think, as I did not do this part of the work.
Okay, please, I thought Dominique Rinaldo is there. Yeah, thank you.
Could you repeat your question, please?
The question is, during the cooking time estimation, did you use the same amount of water? What was the procedure apart from the time? Did you use the same amount of water? Did you vary the dimension of the cubes? I mean, the diameter of the cubes of the yam?
The diameter of the cubes are standardized, and it's cubes with 5 centimeter cubes, and for the boiled yam, we can control the temperature, and the boiling temperature was about 95 degrees, and we waited this temperature before.
Yes, I'm talking about the amount of water. The amount of water. The volume of water that was used to cook.
The volume of water is standardized too, and it is twice the weight of the cube of yam.
Yeah, what I'm trying to say is this. You know, if you call standardized measurement, the dimension, for all the varieties that you check, I suppose that you use the same volume of water for all, because the cooking time can also vary with the water absorption. That's what I'm saying. So ideally, I guess that this is what you do. If you use 50 mils of water for one variety, and you cut the dimension in the same way, so 50 mils of water will be used for all the varieties in order for you to get precise cooking time. Yeah. So it is, those are the things that can vary with the cooking time, because some yam varieties may absorb more water than others. So if you're saying twice the weight, I don't know. If you use the same weight, but it will be very good for you to measure the volume of water used. Even if you are using a yam panda cooker or whatever, you use the same volume of water. So that means the process, everything will be similar. Hello?
Yes, okay, thanks for this remark.
Yeah, thank you. Yeah.
Okay, I have another question for Aliénor. Are you planning to analyze the structure of pectin for raw and cooked yam and plantain?
Yeah, what we would like to look at is more deeply the soluble and non-soluble pectin and their structure using HPSec, for example.
Okay. And you will cook in INRAe?
No, I don't cook in INRAe for now. we already, well, with Dominique Rinaldo, she sent us already cooked products. Why is it nice? Because she cooked them the same way that she characterized the texture. So we know that there's no difference in the cooking process. They all have been cooked by the same person. She did textural analysis, and we do pectin analysis.
Excellent.
Any other comments or question for Aliénor? Agnès Roland-Sabaté, would you like to add something, a precision, some more information, because you've been part of this work as well?
No, thank you for your questions. Just to follow the question of Dominique Dufour, the last question. We will work with as well with Christophe on cooked and on raw plantains. First this year with I can say old plantains samples from Christophe Bugot from Côte d'Ivoire. But after we will work like also with Gérard in Cameroon for plantain. So we will analyze as well raw and cooked plantains.
Okay, because at the moment we have some sample in Côte d'Ivoire. We have RTB food trial there with 36 variety, and I think they are producing now. And if you want to organize some qualification with Côte d'Ivoire, I think it's the moment now. Or maybe during the annual meeting, you will collect some sample to do the work after. Because the plantation is now producing, and they are the clone planted for the RTB food project near Abidjan.
Thank you.
Thank you.
One last question maybe for the team. Last comment. So we can actually close the webinar. Thanks for this presentation, Aliénor. Thanks, Agnès and Dominique, for your contribution. And we all acknowledge the quality of the presentation, and we are looking forward to receiving results of other applications in RTB Foods framework. So thanks a lot for cooperating with other labs. That's really the spirit of RTB Foods. It's cooperation and partnership. So thanks a lot. And we will meet in 2 weeks. At the beginning of December, we will have a presentation from Delphine Ama from IIT in Nigeria. She will be presenting key findings from surveys on plantain products in Nigeria, mainly fried plantain or dodo. She will be presenting a summary of the paper published in the International Journal of Food Science and Technology, recently published and accepted. She will be presenting as well the implication of these key findings for plantain breeders to integrate quality traits in their breeding program. So thanks a lot for joining today, and we will meet in 2 weeks.